Hi everyone, I am Vincent Lara, Application Engineer in Raman Spectroscopy in HORIBA Scientific, and I will present you an introduction to the Multivariate Analysis module in LabSpec. LabSpec 6 now offers the possibility to use new tools which are related to multivariate analysis. These options are available as an optional plugin module. A selected list of algorithms is now available in this module to perform data analysis. The CLS fitting, included as a standard function in LabSpec 6, will not be discussed in this session. But first of all, I would like to show you briefly a couple of examples of the use of NVA illustrating the benefit we could get from it. This mapping is showing the presence of carbon nanotubes spread over a substrate. One is using the monitoring of the intensity of the G-band by the cursors, while the other one is resulting from the MCR treatment of the exact same dataset. This illustrates the advantages that we can get when using NVA in image rendering of a mapping. Another quick example of the benefits of NVA is to help finding out the differences in a dataset. The spectra of these 12 capsules containing the active ingredient as form 1 for 6 of them and as form 2 for the 6 others are pretty much similar at first glance. Clear differences between the two groups can, however, be seen after applying a clustering algorithm showing the ability of Raman spectroscopy to discriminate form 1 from form 2 even within the formulation. Univariate analysis focuses on one single variable, mostly the peak intensity or area or the peak position, to monitor the characteristics of one compound in a mixture. In many cases, this technique is sufficient for simple analysis. However, when the dataset is more complex, with a lot of different constituents or with spectra showing a high number of peaks that may overlap with each other, this technique may show its limitation. On the other hand, multivariate analysis uses several variables simultaneously to have a better description of the data. To give a simple example, it's like studying a given population. Using one variable, for example the age, gives some information about the population, but if you combine that to other variables, for example the weight, the age, the gender, etc., you will get much more information to better describe the population. Various algorithms are available to perform such multivariate analysis. Only the descriptive ones will be described here, decomposition and clustering, but be aware that some of them can be used for quantitative purposes. Let's have a look first at the decomposition principle. The decomposition techniques aim at extracting the main information from a dataset. You can imagine that in a dataset comprising hundreds or thousands of spectra, some of the information will be redundant. For instance, if you record a map of a sample over a substrate, the spectral information due to the substrate will be very much redundant because it is present in every spectrum of the dataset. The decomposition will then isolate the main source of variation of the data induced by the difference of composition in the spectra. As a result, each individual spectrum will be decomposed into a linear combination of new components or loadings, which are often chemically or physically meaningful. Originally, there are as many loadings as spectra in the dataset, each of these loadings containing a part of the variance. The loadings are altered by scores, which are coefficients indicating how much the contribution of each loading is. Practically, only few of these loadings will be enough to well describe the original dataset. The other one, expressing mostly noise, will be discarded. It is quite common that successful decomposition of spectroscopic datasets, including hundreds or thousands of spectra, will involve 10 or less loadings. This number will depend on the complexity of the dataset. To make it clearer, let's take an example. Consider this dataset made of 6 capsules having form 1 as API in blue and 6 other ones having form 2 as API in red. The spectral data indicate that the pattern is common, but small differences are observed. When applying a spectral decomposition, namely PCA, to this dataset, we obtain 12 loadings. Loading 1 accounts for 96% of the variance and looks like the average pattern of the 12 spectra of the dataset. Now, if we look to loading 2, we observe that it shows a kind of spectral structure with some features being positive and other one negatives. This loading expresses only 3.8% of the total variance. Going deeper in loading examination, we observe that loading 6 accounts for only 0.02% of the variance and does not show any interesting spectral feature. This kind of loading is useless and should not be considered. This information is summarized in the table. Only the 2 first loadings account for a non-negligible part of the variance, as they represent 99.8% of the spectral variation and are sufficient to well describe the initial set of 12 spectra. Each original spectrum is the linear combination of the loadings affected by scores or coefficients. For spectrum 1, having form 1, what is remarkable is the negative score of the second loading. On the other hand, when considering spectrum 7, containing form 2, this coefficient is positive. Actually, this second loading traduces the amount of form 2 compared to form 1. Positive coefficient indicates a greater presence of form 2, whereas negative coefficient indicates the opposite. In many cases, the loadings will be meaningful and illustrate any chemical or physical property. The scores can be displayed in a 2D score plot. Each point represents a spectrum and the coordinates of the points correspond to the scores calculated during the decomposition. The 2D score plot of PC1 versus PC2 clearly shows the discrimination of the two groups. This is logical. Spectra having similar characteristics will be projected at the same locations of the score plot because their scores will be quite similar to each other. But be careful. If you do not choose the right principal component, the plot changes dramatically and may become completely meaningless, as in the 2D score plot of PC4 versus PC3. Remember that PC3 and PC4 were negligible and would express only noise. Score plots can be very useful if we want to find evidence of significant differences between groups of spectra. But we need to be very careful in the choice of the PCs. LabSpec 6 NVA module offers the possibility to use 2 different algorithms to perform spectral decomposition, PCA and NCR. Let's look a bit more in details these 2 methods. To show the main characteristics of these 2 algorithms, let's take an example and apply both PCA and MCR to this dataset. This example is a mapping of a pharma tablet comprising around 10,000 spectra. It is known that this tablet contains 3 active ingredients: aspirin, caffeine, and acetaminophen. Let's have a look at the PCA loadings first. The 3 first principal components are displayed, and again, some of them are showing both positive and negative peaks. On the other hand, MCR loadings are looking different in the sense that they show only positive peaks. You may note that PCA1 and MCR3 are looking very much similar, and that many of the features observed in PCA loadings are also visible in the MCR loadings. The score plots are here coded by colors in a similar way as cursor maps, with each pixel corresponding to a spectrum. The higher the score, the more intense is the color. The three score plots shown here correspond to the scores of the three first loadings of the PCA. The MCR score plots are somehow similar to the PCA ones. The main difference is that the score plot of PCA1 is not corresponding to the MCR1, but to the MCR3 one, and vice versa. The table gives indication about variation captured by each loadings. We can note that they are ranked from the most important one to the least important one in the case of the principal component analysis, and that is not true for the MCR decomposition. This is one of the main characteristics of PCA, to rank the principal component according to their importance. Principal component analysis is probably the most popular algorithm to perform decomposition of a given dataset. It involves an orthogonal transformation to generate uncorrelated and independent principal components. Even though the loadings have positive and negative features, they tend to have a chemical meaning. Their examination, and in particular looking at their unique features, help estimating the number of components to keep. This also can be correlated by the variance captured by each loading. When these values become very small, this might correspond only to noise. Contrary to PCA, the loadings obtained by MCR analysis are no longer ranked according to their amount of information captured and are not orthogonal to each other. The algorithm uses some constraints, notably a non-negativity constraint prohibiting the presence of negative features in the components, to improve the interpretability of the loadings. In that sense, MCR aims at recovering pure spectral profiles of the chemical constituents of a mixture, even when no prior information about the mixture is available. As a consequence, the loadings might then be searched directly in spectral libraries for identification. MCR is also involving an iterative algorithm which might be limited and time-consuming if large datasets are analyzed. An example of MCR is given with this map of a geological sample for which no detailed information about the composition was provided. Using the cursor univariate analysis as a first approach gives already some information, but is limited as many different spectra are found, corresponding to mixtures in many cases. The MCR analysis with 8 components leads to the loadings as displayed. The MCR loading 1 is identified as pyrite after library search using the know-it-all package. In a similar way, most of the MCR loadings were identified using the spectral library search, quartz, ilmenite, as well as tectosilicate and inosilicate forms. One question remains, however, open. What is the correct number of components to keep? With PCA, the loadings will be the same whatever the selected number of components. This is not true with MCR. This emphasizes the fact that the choice of the number of components is quite important. Using PCA and crossing the results with MCR might then be recommended to find out an estimation of the number of components to keep. Finally, be aware that not all the loadings might be interpreted, especially if the dataset contains artifacts due to high background fluorescence, etc., etc. It is thus important to apply the necessary preprocessing prior to analysis to remove undesired variability. Let's focus now on the second part, which is the clustering. Clustering is another popular analysis method employing MVA. It is used to group objects having similarities into classes or clusters. It is applied in many different fields, ranging from marketing to the web or to biology. A quite known example is the classification of animal or plant species in biology based on their different properties, physical, morphological, genetic, and so on and so on. In spectroscopy, since the spectra reveal with high specificity the chemical or physical nature of a sample, grouping spectra into clusters according to their similarities will give indications about the different groups of sample included in the dataset. The two algorithms used in the LabSpec MVA module will be explained and illustrated by examples. Hierarchical cluster analysis is an agglomerative technique aiming at grouping objects into clusters. Well, let's take an example with a set of 5 spectra. The spectra are represented here in a 2D plot in a similar way to what is done in principal component analysis plots shown previously. At start, each spectrum is its own cluster. Then, all the distances between the different clusters are calculated to find out which clusters are the closest one. In this example, spectra 1 and 2 are the closest one and are paired to form a new cluster. This can be represented in a dendrogram in which these two spectra are displayed joined together. The distance between these two spectra is reported on the dendrogram, and it is shown by the arrows. The process is then repeated in a new cycle to find out the next closest clusters, spectra 3 and 4 in this example. Again, the distance shown by the arrow corresponds to the one reported on the dendrogram. These cycles are repeated until all clusters are paired, forming then the complete dendrogram. Once the dendrogram is completed, we need to define the different groups or classes. Let's draw a cursor line as in this example. This cursor crosses two lines of the dendrogram, splitting the dataset into two classes. Class 1, comprising samples 1 and 2, and Class 2, made of the 3 other samples. Now, if you move down the cursor, 3 lines of the dendrogram are now crossed, indicating that 3 classes are formed. The number of classes will depend on where the cursor is set, showing more or less details in the clustering. Please bear in mind that the lines between points in the dendrogram correspond to spectral distances, and the greater the lines, the more different are the objects linked by this line. Actually, the hierarchical cluster analysis in the NVA module uses the Ward's algorithm to set the clusters and pair them. It is known to minimize the within cluster variance, that is to say that before grouping one object into a cluster, it simulates what would be the impact of the inclusion of that object on the variance of each cluster, and eventually includes that object into the cluster that undergoes the minimal change of its variance after inclusion. This method implies a large number of calculations, as all distances between each cluster have to be calculated for each cycle of the process, and then it should be restricted to relatively small datasets. Considering a dataset of 1000 spectra, this would represent only for the first cycle 500,000 calculations of distances. Let's take now an example to illustrate these classifications using HCA. Two different people had their hands treated with different products, namely silicone, shampoo, and cream. The hands were washed after treatment. To estimate if the product successfully penetrated in the skin, spectra were taken directly by focusing the beam inside the palm of the hand. The spectra are all looking similar, and the use of MVA is helpful here to find out if any classification into group is possible and down to which level. In LabSpec MVA module, you need to set the number of classes first before running the cluster analysis. When the number of classes is set on 2, the classification shows that the group of silicone-treated spectra, class 1, is clearly separated from the rest. One could have expected that treated skins and clean skins would be the two classes, but the spectral difference induced by the silicone is such that it puts the silicone-treated group apart. Moving up the number of classes to 4 will lead to a more detailed classification, now making the difference between the various skin treatments. Let's now spend a bit of time on divisive cluster analysis. Divisive cluster analysis will group spectra into classes by dividing the space into regions, each region representing one class. To illustrate the DCA, we use the same example with the 5 spectra shown in a 2D plot. In a similar way as HCA, the number of classes is input beforehand. Let's take 3 classes for example. 3 points are then placed now at random positions in the 2D plot. They are shown here in red, blue, and green. Each point is then going to be the center or centroid of a given region, defined in such a way that all the points within that given region are closer to its centroid than to the centroid of any of the other regions. As a result, spectra 1 and 2 are in the blue region, spectrum 3 in the red one, and spectra 4 and 5 in the green one. During the next step, the centroids will move to become the mean of the new points included in each region. Since the centroids moved, the divisions of the space have to be reset to take into account these changes. The limits of each region are then redefined according to the same rule as the one applied in the previous step. The picture changes quite dramatically, and now the spectrum 4 that was included in the green region is now part of the red one. Again, the centroids move so as to become the mean of the points included in each region. This whole process of space division is repeated until the assignment of samples in the groups no longer changes. The 3 classes are then set, grouping the spectra according to their similarity. Class 1, the blue one, includes sample 1 and 2. Class 2, sample 3 and 4. And class 3, only sample 5. The algorithm used in DCA is known as the k-means algorithm. It includes two steps: the step of assignment of every single object or spectrum in the cluster whose mean is closest to it, and a step of recalculation of the new means, the centroids. These cycles are repeated until no change in assignments is observed. It is requiring fewer calculations than the HCA, as only distances between single spectra and centroids are calculated, and not interspectral distances. This method is therefore much more suited for the analysis of large datasets and particularly mappings. The mapping of the pharmaceutical tablet is used to illustrate the DCA clustering. Three classes are set corresponding to the three compounds of the tablet: acetaminophen, aspirin, and caffeine. Three maps are displayed, each color representing a class. This picture is somehow similar to the maps obtained with MCR and PCA, but the difference here is that each pixel is uniquely assigned to a class. The statistics table accompanying the maps displays the number of pixels or spectra included in each class. Please be aware that these values cannot be used for quantitative purposes, although they may give interesting semi-quantitative information about the composition of the sample. The number of classes is important. If 4 classes are input, then the final result is dramatically changed. The green map, mostly surrounding the red particles of acetaminophen, represents the mixture acetaminophen-aspirin and not a pure compound. An important step of the multivariate analysis involves the data preprocessing. Let's have a look at the different preprocessings and their use. The goal of data preprocessing is to correct for some undesired variance. For example, if you map a sample in which the background fluctuates upon the measurement positions, you will generate spectra with different background, thus adding some undesired information to the spectra. Using a preprocessing to remove the background is then recommended prior to running the NVA treatment, so as to focus only on the meaningful information lying in the spectra. Some of these preprocessings are included in the NVA module: normalization, mean centering, and autoscale. Normalization is very common and compensates for differences in intensity induced, for example, by laser power drifts or differences in focusing, and is applicable almost to any dataset. Be aware that some of these preprocessings really alter the original data. This means that the loadings calculated after applying these preprocessings would be little or not interpretable. This is the case of mean centering or autoscale, involving a step of mean subtraction. Here is an example of a dataset. You may recognize the spectra of the 12 capsules, displayed after application of various preprocessings. You may notice how important can be the modification of the original dataset. Also, some of them produce negative features which make them not compatible with MCR algorithm. There is no specific rule regarding the use of the different preprocessings, but just few recommendations. Normalization should be preceded by some background removal function first, not to include the background information in the normalized data. Mean centering is rather well suited when you have similar data, as it will remove the general pattern from the spectra and focus only on the small differences. Data preprocessing can be combined to add their effects. In addition, other preprocessings which are not included in the NVA module but available in the Processing tab of LabSpec 6 can be employed. This is the case for baseline correction or derivation, both of them used to remove high spectral background. Derivation is also a way to enhance spectral differences. For example, shoulders present in the original data will turn into peaks after derivation. Other normalizations are available, such as the normalization to Multivariate, which is very much employed for spectroscopic data. I would also like to draw your attention on another type of preprocessing, the selection of the spectral range. You may get benefit of reducing the spectral range to focus only on interesting parts or simply to remove saturated parts that would bring useless information. It is also sometimes useful to cut the spectral data to reduce the calculation. To prove the usefulness of data preprocessing, let's have a look to the results of the clustering example with and without preprocessing. The picture dramatically changes and the expected classification is no longer observed. The conclusions of the experiment might have been completely wrong if no preprocessing had been applied to the data. In that case, you can easily imagine that the measurement of the palms were not very much reproducible, thus leading to increased variation induced by the measurement itself. I would then encourage you to use these preprocessings before applying any multivariate treatment. Well, I hope this presentation helped you having a better idea of the use of the NVA methods and the benefits we can get from them. It does help a lot, and sometimes it is the only way to utilize the data properly to get the desired results. But it is not a black box that you should use without taking care and asking yourself a number of questions first. You should always keep an eye on the data, as they always contain all the fundamental information, and also try to confirm the results with other methods whenever possible. Check the HORIBA scientific website to get the latest news and updates about Raman spectroscopy. Thank you very much for listening.